A very warm welcome to Cullen Platform Igniting Curious Mind has on date the 6th June 2026. Chapter 8, The Cell, the Unit of Life. You look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, and what is it that inanimate things that does not have, which a living thing has. The answer to this is the presence of basic units of life. The cells is all living the cell is in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular, like while the others composed of many cells are called as multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence, perform essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is a fundamental structural and functional unit of all living organisms. Antony von Leeuwenhoek was first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of microscopes and its improvement leading to the electron microscope reveals all the structural details of a cell. Cell theory. In 1839, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells and from the tissues and plants of plants. At the same time, the Theodor Schwanz, a German nerve zoologist, studied different types of animal cells, reported that the cells had a thin outer layer which is today known as the plasma membrane. He concluded, based on his studies on the plant tissues, that the presence of a cell wall is a unique characteristic of the plant cells. On this basis, the Schwann proposed a hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory, which cell theory, however, did not explain as to how the cells were formed. Rudolf Wischer first explained that cell divides and new cells are formed. Pre-existing cells from Onimus cellulose e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. The cell theory is understood today as all living organisms are composed of cells and the products of cells. All cells arises from the pre-existing cells. An overview of a cell. You have earlier observed cells is an onion in an onion peel and a human cheek cells under microscope. Let us recollect their structure. The onion cells is a typical plant cells, is a distinct cell wall and its outer boundary is just within its cell membrane, and the cells of the human cheek have an outer membrane and delimiting structures of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains chromosome, which in turn contains genetic material, DNA. The cells that have a membrane bound nucleus are called as eukaryotic, whereas cells that lack a membrane bound nucleus are called prokaryotic. In both the eukaryotic and prokaryotic, a semi-fluid matrix called cytoplasm occupies a volume of the cell. The cytoplasm is a main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside nucleus, the eukaryotic cells have both other membrane bound distinct structure called as organelles, like the endoplasmic reticulum, a Golgi complex, lysosome, mitochondria, microbiotes, vacuoles. Prokaryotic cells lack the membrane bound organelle. Ribosomes are non-membrane bound organelle found in all the cells, both eukaryotes as well as prokaryotic. Within the cell, ribosomes are not only found in cytoplasm but also within the two organelle, chloroplast in plant cells and mitochondria or unrough ER. Animal cells contains non-membrane bound organelles called centrosomes, which helps in the cell division. The cell differ greatly in size, shape, and activities. For example, the mycoplasm, a smallest cells and 0.3 mm in length, while bacteria, which is 3 to 5 mm, and the Largest isolated single cell in the egg of an ostrich. The multicellular organism membranum human red blood cells are about 7.0 million in diameter, when your nerve cells are some of the longest cells. The cells also vary greatly in their shapes and may be a disc-like, polygonal, or columnar, cuboidal, and thread-like, or even irregular. The shape of the cells may vary with the functions of them. They perform. A prokaryotic cell. The prokaryotic cells are represented by the bacteria, the blue-green algae, and the mycoplasma pleuropneumonia like organism. They are generally small and multiple more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus rod-like, cocosperical, vibrocoma shaped, and spirulum. The organizations of prokaryotic cell is fundamentally similar, even though prokaryotic exhibits a wide variety of shapes and functions. All the prokaryotic have a cell wall surrounding the cell membrane except in the mycoplasm, the semi-fluid matrix filling the cell. In the cytoplasm, there is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition, the genomic DNA, the single chromosome, and circular DNA. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called as plasmids. The plasmids contain plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that the plasmids Plus, this plasmid DNA is used in monitoring bacterial transformation with a foreign DNA. A nuclear membrane is found in eukaryotes. Non-organelles like the one in the eukaryotes are found in prokaryotic cells except the ribosomes. Prokaryotes something unique in the form of inclusion. A specialized differentiated form of cell membrane called as mesosome is a characteristics of the prokaryotes. They are essential in flooring of the cell membrane, the cell envelope, and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemical complex cell envelope. The cell envelope consists of a tightly bound three-layer structure. The outer is a glycocalyx followed by a cell wall and then a plasma membrane. Although each layer of envelope perform a distinct function, they act as together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelope and the manner in which they respond to the staining procedure developed by the grams. Those that take up the gram strains are gram positive, and those that do not is gram negative bacteria. Glycolic is different compositions and thickness among the different bacteria. It could be loose sheets called as slime layer in some, while in other it may be thick and tough called as capsule. The cell wall determines the shape of the cells and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature, interact with the outside wall. The membrane is similar in structurally and to that of a eukaryote. A special membrane structure is mesosome, which is formed of an extension of a plasma membrane into the cell. The extensions are in the form of vesicle, tubules, and lamellae. They help in the cell wall formation, DNA replication, distributions to the daughter cells. They also help in respiration, secretion process to increase the surface area, the plasma membrane, and enzymatic content. In some prokaryotes, like the cyanobacteria, there are other membrane extensions into the cytoplasm, that is chromatophores, which contains pigments. Bacterial cells may be motile, non-motile. If motile, they have a thin filamentous extensions from the cell wall called flagella. The bacteria show a range of numbers in arrangement of flagella. Bacterial flagellum is composed of three parts: filaments, hooks, and the basal body. The filament is a longest portion extends from the cell surface to the outside. Beside flagella, pillae fimbriae are also the surface structure of the bacteria do not play a role in motility. The pillae is a elongated tubular structure made up of special protein. The fimbriae is a small bristle-like fiber sprouting out of the cells. In some bacteria, they are known to help attach the bacteria to rock in the stems and also to the to the host tissues, ribosome and the inclusions body. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They're also 15 nanometer to 20 nanometer in size and made up of two subunits, 50 years and 30 years. Units which then present together has a 70 years. Prokaryotic ribosomes are site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called as polyribosome or polysomes. Ribosomes or polysomes translates the mRNA into protein. Inclusions body. The reserved material in the prokaryotic cells are stored in the cytoplasm in the form of an inclusion body, and these are not bound by any membranous system and lie free in the cytoplasm. The phosphate granules and the granulophyce granules and the Granophyce granule lie free in the cytoplasm. The phosphate granules and Synophyce granules and the glycogen granules and the gas vacuoles are found in the blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells, eukaryotes includes all the protist plants and animals and fungi. In the eukaryotic cell, there is an extensive compartmentalization of a cytoplasms. Though the presence of membrane-bound organelles, eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. This genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plants and animal cells are different as they possess cell wall and plastids and large central vacuoles which are absent in the animal cells. On the other hand, animal cells have a centrioles which are absent in almost all plant cells. Now, for diagrams of plants and animal cells, let us now look at individual or cell organelles to understand their structures and functions. Cell membrane. Detailed structures of the membrane have been studied and only after advent of electron microscopes in 1950s. Meanwhile, a chemical study on the cell membrane, especially in the human blood cells, enabled the scientists to deduce the possible structures of plasma membrane. These structures shows that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer and also lipids that are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tail towards the inner parts. This ensures that the non-polar tails of saturated hydrocarbon is protected from the aqueous environment. In addition to phospholipids, the membrane also contains cholesterol. A later biomolecular investigations clearly revealed that the cell membrane is also possess protein, carbohydrates. The ratio of protein and lipids varies considerably in different cell type. In human being, the member of the erythrocytes has approximately 52% protein and 40% of lipids. Depending on the ease of extraction, membrane proteins can also be classified as integral and peripheral. The peripheral protein lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. The fluid mosaic model of the plasma membrane. An improved model of the structures of a cell membrane was proposed by Singers and Nicholson in 1972, widely accepted as fluid mosaic models. According to this fluid mosaic model of a lipid enable lateral movement of a protein within the overlap bilayer. This ability to move within the membrane is measurable as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like the cell growth, formation of intracellular junctions, secretions, endocytosis, and cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some of the molecules present on either side of it, while many other molecules can move briefly across the membrane without any requirement of energy, and this is called as passive transport. The neutral solutes move across the membrane by a process of simple diffusion along the concentration gradient from a higher concentration to lower. Water may also move across this membrane from a higher to lower concentration. Movement of water by the diffusion is osmosis, as per the polar molecules cannot pass through a non-polar lipid bilayer. They require a certain carrier proteins of the membrane to facilitate their transports across the membrane. A few of a molecule are transported across a membrane against their concentration gradient, and the lower to a higher concentration gradient. Such a transport in the energy dependent on the process in which ATP is utilized, and it is active transports Na plus and K plus pump. Cell wall. As you recall, a non-living rigid structure of a cell wall forms the outer covering of a plasma membrane of fungi plants, and the cell wall not only give shapes to cells and protect the cell from the mechanical damage and infection, it also helps the cells to cell interact, provides barrier to undesirable macromolecules. Algae have a cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonates, while in other plants it consists of cellulose, hemicellulose, pectin, and protein. The cell wall of a young plant cell's primary wall is capable of growth, which gradually diminishes as the cells mature, and the secondary wall is formed of a inner towards the membrane sides of the cell. The middle lamellae is a layer mainly calcium pectate, which holds or glues different neighboring the cells together. The cell wall on the middle lamellae may be traversed by the plasmodesmata, which connect the cytoplasm of the neighboring cells. The endomembrane system. While each of the membrane organelles is distinct in terms of its structures and functions, many of these are considered as an endodermal system because their functions are coordinated. The endomembrane systems is endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Since the function of the mitochondria, chloroplasts, and perisomes are not coordinated with the above components, they are not considered as a part of endomembrane systems. The endoplasmic reticulum. Electron microscope studied of eukaryotic cells reveal the presence of network of a reticulate of teeny tubular structures scattered in the cytoplasm called endoplasmic reticulum. Divides the intracellular space into two distinct components: luminal inside extraluminal cytoplasm component. Often show a ribosome attached to outer surface. Endoplasmic reticulum bears a ribosome on the surface is called rough endoplasmic reticulum. In the absence of a ribosome, they appear smooth and are called a smooth endoplasmic reticulum. is frequently observed in the cells actively involved in protein synthesis and secretions. They are extensive and continuous with the outer membrane of the nucleus. Smooth endoplasmic reticulum is a major site for synthesis of lipids. In animal cells, the lipids like the steroidal hormones are synthesized in SCR. Golgi apparatus. The chameleon Golgi in 1898 first observed a dense range of reticulate structures near the nucleus. These were named as Golgi bodies after him, consist of a flat disc-shaped sac or crystal of 0.5 mm to 1 mm diameter. These are stacked parallel to each other. Varied numbers of cristae are present in Golgi complex. The Golgi cristae are concentrately arranged near the nucleus with a distinct convexes or forming a phase that concave trans or the maturing phases. The cis and trans phases are the organelles which entirely different from that but interconnected. The Golgi apparatus principally perform the function of packaging materials and to be delivered either to the intracellular targets or secreted outside the cells. The material to the packaged in the form of vesicles from the refuses the cis phases of Golgi apparatus and move towards the maturing phases. This explains why Golgi apparatus remains in the close associations with the endoplasmic reticulum. A number of protein synthesis of ribosomes on the endoplasmic reticulums are modified in the cristae of the Golgi apparatus before they are released from its trans phases. Golgi apparatus is important inside the formations of a glycoproteins and glycolipids. Lysosomes. These are the membrane-bound vesicle structure formed by the process of packaging in the Golgi apparatus. Isolated lysosome vesicles have been found to be very rich in almost all types of hydrolytic enzymes. The hydrolase, lipase, protease, and carbohydrates optically active at the time of acidic pH. These enzymes are capable of digesting carbohydrates, protein, lipids, and nucleic acids. Vacuole. Vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other material that are not useful for the cells. The vacuole is bound by a single membrane called a stonoblast, and the plant cell vacuoles can occupy 90% of volumes of the cells. In plant, the tonoplasts facilitate the transport of numbers of ions and other materials against the concentrations gradient into the vacuoles. Hence, their concentration is significantly higher in the vacuoles than in the cytoplasm. In amoeba, the contractile vacuoles is important in osmoregulation and excretion. Many cells, as in the protista food vacuoles, are formed by engulfing food particles. Mitochondria. Mitochondria is unless specifically stained, not easily visible under the microscope. The number of mitochondria per varies depending on the physiological activity of the cells. In terms of shape size, considering the degree of variability in observations, typically it is a sausage shaped, cylindrical, having a diameter of 1 mm average to zero point 0.5 mm and the length of 1.0 to 4 mm. Either mitochondria is a double membrane bound structure with outer membrane and inner membrane dividing the lumen distinctly into two aqueous compartments, an outer compartment and inner compartment. And the inner compartment is filled with the dense homogeneous substances called as matrix, and the outer compartment is a continuous lining boundary of organelles. The inner membrane forms a number of infloring called as cristae towards a matrix, and the cristae increases surface area. The two membranes have their own specific enzyme associated with the mitochondrial functions. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called as powerhouse of the cells. A matrix also possesses single cellular DNA molecules and a few RNA molecules, ribosomes, 70S, and the component required for synthesis of protein. The mitochondrial divide by the fissions. Plastids are found in almost all plant cells and euglenoids. These are easily observed under the microscope as they are large and bear some specific pigment and imparting specific color to the plants. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, leucoplast. So chloroplast contains chlorophyll, carotenoid pigment, which is responsible for trapping light energy essentially for photosynthesis. In the chromoplast, the fat molecules, carotenoid pigments like the carotene, xanthophylls, and other present. This gives a part of the plant yellow, orange, red color. The leucoplasts are the colorless plastids, vary shape, size, and store nutrient. Amyloplast stores carbohydrate starch. The potato Elioplast stores oils and fats, whereas aleuroplast stores proteins. Majority of the chloroplasts of green plants are found in the mesophyll cells of the leaves, and these are lens-shaped, oval, spherical, discoid, even ribbon-like organelles having a variable length, 5 to 10 mm, width of 2 to 5 mm, and the number varies from 1 per cell. The chlamydomonas has in green algae to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are double membrane bound on two inner chloroplast membrane is relatively less permeable. The space limiting by inner membrane of the chloroplast is a stroma, and the number of organized flattened membrane sac called as thylakoids present in the stroma. The thylakoids are arranged in stalk-like piles of coins called as grana and intergranular thylakoids. In addition, there are flat membranous tubule called as stroma lamellae connecting the thylakoids of different grana. The membrane of thylakoid encloses space called as lumen. Stroma of a chloroplast contains enzyme required for synthesis of carbohydrates and proteins. It contains a smaller double circular DNA molecular ribosome. Chlorophyll pigments are present in thylakoids. The ribosomes of the chloroplast are 70S, and then the cytoplasmic ribosome are 80S. Ribosomes are the granular structure observed under the electron microscope and dense particles by the George Balde 1953. They are composed of ribonucleic acid RNA and the proteins and are not surrounded by any other membrane. Eukaryotic ribosome is 80S, while the prokaryotic ribosome is 70S. Each ribosome has two subunits, larger subunits, and the two subunits of 80S ribosomes are 60 and 40S, while that of a 70S ribosomes are 50S and 30S. Here, S is a SEDWORKS unit stands for the sedimentation coefficient. It is indirectly a measure of density and sizes. Both 70S and 80S are composed of two subunits, cytoskeleton, an elaborate network of filaments, proteinaceous structure consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm, collectively referred to as cytoskeleton. The cytoskeleton is a cell that involved in many functions such as mechanical support, motility, maintenance of the shapes of the cells. Cell and cilia and the flagella. Cilia and flagella are the hair-like outgrowth of a cell membrane. The cilia are the small structure which work like a and causing a movement of either of the cell on the surrounding fluid. Flagella is a comparatively longer and responsive cell movement. Prokaryotic bacteria possess flagella, but these structurally different from the other eukaryotic flagella. Electron microscopic study of cilium and flagella show that they are covered with a plasma membrane. The core is called as annexomere. The possessing a number of microtubules running parallel to the long axis, the annexomere usually Axoneme usually have a 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubule is referred to as 9 plus 2 array and array. The central tubules are connected by a bridge and is also enclosed by a central sheath which is connected to one of the tubules of the peripheral doublets of the radial stalks, and these are the 9 radial spokes. The peripheral doublets are interconnected by the linker. Both the cilium and the flagellum emerge into a centriole like the structure called as basal body, centrosomes and centrioles. Centrosomes are organelles usually containing two cylindrical structures called as centrioles. These are surrounded by anonymous amorphous pericentriolar material. Both the centrioles in a centrosomes lie perpendicular to each other, in which each has organizations like the cartwheel. They are made up of 9 even spherical fibril and tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal regions of the centriole is also a proteinaceous called as hub, which are connected with the tubules of the peripheral triplets of radial spokes made up of protein. The centrioles form the basals, body of the cilia and the flagella, spindle fiber that give rise to spindle apparatus during the cell division in the animal cells. Nucleus is a cell organelle that first described by the Robert Brown and early 1831. Later material of the nucleus trained by the basic dyes was given by the name chromatin by the Fleming. The interface of a nucleus of a cell which is not divided, has highly extended and elaborate nucleoprotein fiber called as chromatins, a nuclear matrix, and one or more spherical body called as nucleoli. The electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes, have a space between 10 to 15 nanometer as perinuclear spaces. Before forms a small barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears a ribosomes on it. At the time of a number of places, the nuclear envelope is interrupted by a minute pores, which are formed by the fusion of two membranes. These nuclear pores are of passage through which the movement of RNA and protein molecules takes place in both the directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell, and variation in the number of nuclei are also frequently observed. Can you recollect the name of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Erythrocytes of many of the mammals receives sieve tube cells of the vascular plants. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contains nucleus and the chromatin. The nucleoli are the spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm, as it is not a membrane-bound structure. It's a site for the active ribosomal RNA synthesis. Large and more numerous nuclei are present in the cell activity carrying out the protein synthesis. You may recall that the interface of the nucleus has a loose and distinct network of nucleoprotein fiber called as chromatin, but during different stage of cell division, cells shows a constructed chromosome in place of nucleus. The chromatin contains DNA and some basic proteins called as histones, non-histone proteins, and also RNA. A single human cell had approximately 2 meter long thread of DNA distributed along its 46, that is 23 pairs chromosomes. You may have studied the detail of DNA packaging in the form of chromosome in the form in class 12. Every chromosome visible only in dividing cells essentially have a primary constructed or a centroplasmic or based on the positions. The based centromes holds two chroma sorry Every chromosome visible only during the dividing cells essentially have a primary constructions on the centromere, on the sides of which a disc-shaped structure called as kinetochores are present. The centrosomes holds the true chromatids of the chromosomes based on the position of the centromere. The chromosomes can be classified into four types. The metacentric chromosome has a middle centrosome forming two equal arms and the of the arm of the chromosome. The submetacentric chromosomes are has a centromere slightly away from the middle of the chromosomes, resulting into two shorter and longer arms. In case of acrocentric chromosome, the centromere is situated close to its end, forming one of the extremely short and very long arm, whereas the heterocentric chromosomes has a terminal centromere. Sometimes a few chromosomes are non-strained secondary constructions at the constant location give the appearance of a small fragments called as satellites, microbodies. Many members of the bound minute vesicles called microbodies that contains various enzymes present in both plants and animal cells. Summary: all the organisms are made up of cells and aggregates of cells. Cell vary in their shape, size, and activities and the functions based on the presence and absence of the membrane bound nucleus and other organelle cells, and hence called cells. Hence organisms can be named as eukaryotic and prokaryotic. A typical eukaryotic cell consists of a cell membrane, a nucleus, and cytoplasm. The plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable, facilitate transports of several molecules. Endomembrane system, that is Golgi body complex, lysosome, and vacuoles. All the cell organelles perform different but specific functions. The centrosomes and the centrioles form the basal body of the cilia and flagella and facilitate the locomotion. In animal cell, the centrioles also forms a spindle apparatus during the cell division. The nucleus contains a nucleoli, the chromatin network. It not only controls the activity of organelle but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cristae, and there are two types of rough and smooth. And the ER helps in transport of substance, the synthesis of protein, lipoproteins, glycogens. A Golgi body is a membrane organelle composed of a flattened sacs and secretion of cells and are packed in them and transported from the cells. The lysosome are a single membrane structure containing enzymes for the digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm and are associated with the ER. The mitochondria help in oxidative phosphorylations and generation of adenosine triphosphate, and they are bound by the double membrane. An outer membrane is smooth and inner one is folded into several cristae. The plastids are the pigment containing organelles found in the plant cells only. In the plant cells, chloroplasts are responsible for trapping light energy essentially for the photosynthesis. The grana is the plastid is a site of light reaction and the stroma of a dark reaction. The green colored plastids are chloroplast, which contains chlorophyll, whereas the other colored plastids are chromoplast, and they may contain pigments like the carotene xanthophylls. The nucleus is enclosed in a nuclear envelope and double membrane structure within the nuclear proteins. The inner membrane encloses the nucleoplasm and the chromatin material, and this cell is a structural and functional unit of life. Thank you for watching. You're tuned into current platform Igniting Curious Mind, where you and I grow together.